Now that we've discussed the structure of blood vessels, let's take a closer look at the different types of blood vessels and what they're particularly important for. Let's start with the arteries. The arteries have the thickest, most muscular, and most elastic blood vessel wall. They have a lot of layers of smooth muscle in the tunica media, and there are also a lot of elastic fibers in the tunica media. Not only are the arteries responsible for carrying high pressure blood away from the heart to distribute it to all the areas of the body, the arteries are also important for reducing the blood pressure. It's important to reduce the blood pressure to avoid stress on the smaller blood vessels. In order to reduce the blood pressure, the arteries expand and then recoil with each pulse, and that starts to absorb some of the force of the blood pushing through the arteries. Here you can see a diagram showing the high blood pressure in the aorta where the blood is first coming out of the heart, and you can also see the big change in pressure between the systolic pressure when the heart is pumping and the diastolic pressure when it's at rest. So not only do we have a high pressure, but it also pulses, which could be very damaging to small blood vessels. The arteries help reduce this, but we actually see a more dramatic result from the arterioles. The arterioles are the smallest of the arteries. They only have a few layers of smooth muscle in their blood vessel wall, and they play a very important role in reducing the blood pressure. And this is going to prevent damage to the capillaries that could easily rupture if they were experiencing the sort of pulsing of the blood pressure that we see in the arteries. So here where the arterioles are, you can see we drop the blood pressure dramatically, and we go from having pulses of pressure to having a smoother pressure that won't damage the capillaries. In addition to regulating the blood pressure and reducing it as it goes into the capillaries, the arterioles are also important for regulating the blood flow to specific areas of the body. We can control the diameter of the arterioles, make the arterioles wider to carry more blood to certain areas of the body, or we could make the diameter narrower to carry less blood to a particular area of the body. But there's a more specific way that arterioles can control the blood flow to and from certain areas, and that is through the use of special little circles of muscles called precapillary sphincters. If you take a look at this picture, on this side we have an arteriole. This is going to be carrying oxygenated blood from the heart toward a capillary bed. So here's a bed of capillaries here. Running through the capillary bed is the smallest type of arteriole called a meta-arteriole. And where the meta-arteriole meets the capillaries, we have a small circle of smooth muscle called a precapillary sphincter. And this precapillary sphincter is going to control whether blood is allowed into the capillary bed or not. When the precapillary sphincter is open, if it's relaxed, then blood can flow from the meta-arteriole into the capillaries and carry blood to all the cells served by that capillary bed. If the precapillary sphincters are closed, if they're contracted, then no blood will be able to go from the meta-arteriole into the capillary bed, and we won't be getting blood to that particular area of tissue. It's important to note that we don't actually have blood going to all areas of the tissue all the time. We simply don't have enough blood in our body to serve all those different capillaries all at once. So instead, we open up the precapillary sphincters to certain capillary beds so that we get flow of blood to those capillary beds, and once we've had enough nutrients and oxygen to those cells, we'll close that off and open the precapillary sphincters to a different capillary bed so those cells can get nutrients and oxygen, and then we close those off and we can reopen another. So by circulating where the blood can go, we can get by with less blood volume and still maintain an adequate amount of nutrients and oxygen going to all of the cells. Now there are some specific signals that tell the arterioles when a certain tissue needs more blood flow and when you really should open up the precapillary sphincters to let some more blood in. So the muscles of the precapillary sphincter are sensitive to things like low levels of oxygen and high carbon dioxide. If a tissue has low oxygen or high carbon dioxide, that's a signal that it needs more blood flow. That causes the precapillary sphincters to open and blood can flow into those capillary beds. An increase in acid or a decrease in pH is another signal that a tissue needs more blood flow. Acids are produced as a tissue is working or producing or functioning, and so high acid means the tissue has more need for oxygen. Finally, increased temperature in a tissue also indicates the need for more blood flow. So any of those things, low oxygen, high CO2, low pH, or higher temperature will cause precapillary sphincters in an area of the tissue to open to bring more blood to the area. It is important to note that we're talking about specific small areas of tissue here, not a big change through the whole body, but just one small specific area of tissue. If it gets low oxygen and needs a little bit more, we can open precapillary sphincters to bring more blood flow to that particular area. Let's talk a bit about capillaries, the smallest of the blood vessels. On average, they're only about 5 to 9 micrometers in diameter, although the smallest ones can be only 3 or 4. In this picture, you can see a capillary running along a muscle cell, and you can see it's so small that we can only fit a single file row of red blood cells running through this capillary. The really important function of capillaries is the exchange of materials. Only in the capillaries can we get materials out of the blood vessels and out into the tissues and get wastes and things from the tissues back into the blood vessels to be transported around the body. So exchange of materials is vitally important, and that happens in the capillaries. If you remember what we talked about at the beginning of the semester, the main forces involved here are the blood pressure or the hydrostatic pressure that pushes fluids and molecules out of the capillaries into the tissue fluid, and then the osmotic pressure inside the capillaries that's pulling fluids back from the tissue into the blood vessel. Some things are able to actually diffuse across the endothelial cells that make up the capillaries to get in and out of the capillaries. Most things, however, filter out between the cells or through gaps in the cells. And this brings us to 3 different types of capillaries that allow the exchange of different types of materials. The most common type of capillaries are called continuous capillaries. These are found in most tissues of the body. It's made up of a single layer of endothelial cells, and these endothelial cells are held together by tight junctions, but they have some little clefts in between them. There's an intercellular cleft between the cells. This intercellular cleft is about 4 nanometers in diameter, so it's very, very tiny, and it allows things to filter out between those capillary cells. Only very small things can get out, things like water, glucose, amino acids. Big things like proteins are not going to be able to squeeze out through that tiny intercellular cleft. The second type of capillaries are the fenestrated capillaries, shown here. The fenestrated capillaries are similar to the continuous capillaries, only they have fenestrations or filtration pores that go through the epithelial cell. So it's almost like a little hole through the middle of a donut, a hole through the epithelial cell that allows materials to get in and out of the capillary more easily. These fenestrations or filtration pores are between 20 and 100 nanometers, so they're a lot bigger than the intercellular clefts that we see in the continuous capillaries. So we can get bigger things in and out. We can also get larger volumes of things in and out. So we find these fenestrated capillaries in areas of the body where we need better exchange of materials. Particularly, we find them in the digestive system, in the small intestine, where you're trying to absorb a large amount of nutrients. And we also find them in the kidneys, where you're trying to get rid of a lot of waste molecules. So the nutrients and waste are able to get in and out of the capillaries easier if we've got fenestrated capillaries. The third type of capillary is interesting. Sometimes it's not even called a capillary, but it's called a sinusoid or sinusoid capillaries. These are found only in a couple areas of the body, including in the liver and in the bone marrow. And these capillaries are better described as being sort of larger blood-filled spaces. The endothelium of these sinusoids is not continuous. They're actually gaps or spaces between the cells where they don't come right together. The advantage to this is that it leaves big gaps for things to get in and out of the capillaries. That's why we find these in the liver and in the bone marrow. In the bone marrow, we have to have spaces where we can get blood cells that are too big to get in and out of capillaries into circulation. So in the bone marrow, the red blood cells can get into the capillaries through these sinusoid capillaries. And in the liver, where we're making tons of big plasma proteins that are going to be too big to get through even the filtration pores and fenestrated capillaries, they can get into circulation through these sinusoid capillaries. Once we've completed the exchange of materials between the capillaries and the tissues, then the blood that is now given up a lot of nutrients and oxygen and is full of wastes needs to be collected and sent back to the heart. And it's collected in venules. So here we can see the capillary bed is feeding into this blood vessel, carrying it to a venule. Venules are the smallest veins. They have very thin walls. And in addition to collecting the blood coming from the capillaries, there can be some small amount of exchange across the venules because they do have such very thin walls. The venules carry the blood to the veins. If we take a look at veins, they have a thinner wall than arteries, fewer layers of smooth muscle, and a lot of connective tissue. The other really important thing about a vein is that veins contain valves. These valves are really important to keep the blood moving the correct direction. In the arteries, we don't need valves because the blood pressure keeps the blood flowing in the correct direction. In the veins, the blood has a very low pressure, and so it would fall back down to our feet by gravity if we didn't have valves to keep the blood moving along. When we talk about the functions of the veins, there are two main functions. One is that the veins actually serve as a reservoir for our blood. More of our blood is found in our veins than anywhere else in the body. So in this chart from your book, you can see that at any one time we have about 12% of our blood in our heart and about 18% of our blood going somewhere to our lungs. The rest of the blood is in systemic circulation, either arteries or capillaries or mostly in the veins. So at any given time, about half of your blood is actually being held in your veins. In addition to holding your blood, your veins need to get the low pressure blood back up to your heart. And this is tricky because, as we said, it's low pressure. So there are a number of things that help to move the blood through the veins toward the heart. First is the pressure. There is a little bit of pressure in the veins. From this you can see that there's not nearly as much pressure in the veins as there is in the arteries, but there is still a little, and that does help move the blood in the correct direction. A second factor that helps move blood through the veins back toward the heart is gravity, which works really well for the top half of the body, especially if you're standing up. Doesn't work as well for the lower half to try and move the blood up. But when you lay down, especially if you lay down and put your feet up, then gravity is working to help return that blood back to your heart. Another really important mechanism for returning blood to the heart is skeletal muscle contraction. Whenever your muscles contract, they squeeze your veins. They squeeze the veins running through those muscles. And that pressure pushes the blood. And because the valves are preventing the blood from going backwards, it pushes the blood toward the heart. So the more you contract your muscles, the more blood you are returning to your heart. Which is why it's important to move around to ensure good circulation. The last mechanism for moving blood back toward the heart is simple breathing. Whenever you breathe and you expand your lungs, that puts pressure on the vena cava. So when you compress the vena cava, that also is going to push blood toward the heart, since the valves are going to keep it from going backwards. So between the blood pressure, the effects of gravity, skeletal muscle contraction, and breathing, we're able to keep blood moving toward our heart, so blood gets from our veins back to our heart, so we can push it out to our arteries again.